So today our topic is tuberculosis, and the figure represents some of the mechanisms of the disease transmission and which part is affected, whatever the bacteria that cause the tuberculosis disease. So I want to know how many of you have heard of the tuberculosis and what do you know about it? So many of the students have raised their hands that they know about the tuberculosis and they also know that it is a respiratory disease that causes that infect the lungs. Thank you, and it is appreciated. So learning objectives. So at the end of the chapter, we'll know about or we can discuss about the general properties of the etiological agents, describe the pathogenesis and the disease caused by them, and identify the clinical features of the disease. So these topics will be covered in this tuberculosis. So let us know. So another, which organs are most commonly affected by TB? And how many you have been met with someone diagnosed with the TB? So most of the students have raised their hands and said that the lungs are affected majorly, and the few students have also raised their hands. They have met to the persons who have diagnosed with the TB. So let us discuss about the general introduction, that is the mycobacteria. These are the group of the mycobacterium. So what are the features or what are the morphological structures of this bacteria have? So let us discuss about that one. So the bacteria that belongs to the group mycobacteria are rod-shaped. They are rod-shaped, aerobic in nature, and non-spore forming. And they also resist the decolorization by acid or the alcohol. So it is known as the acid-fast bacilli. So it is based upon the name that the staining is done, that the Zn staining, on the basis of the Zn stain. Because when we use the carbon 14 for the staining, the carbon 14 is not decolorized once it take the stain. So that it is known as the acid-fast bacilli, because the decolorizer that contains the acid alcohol composition. By that way, it is named as a acid-fast bacilli. And very important pathogens of this mycobacteria are Mycobacterium tuberculosis, that causes the tuberculosis, and the Mycobacterium leprae, that causes the leprosy. So let us discuss about the etiological agent, that is the Mycobacterium tuberculosis. So particularly, just before we have discussed about the group, that is the mycobacteria. Now we are focusing to the Mycobacterium tuberculosis that causes the disease known as the tuberculosis. So what type of bacteria is this? As a general characters we have discussed, but this particularly or specifically what kind of the bacteria is the Mycobacterium tuberculosis? It is a thin, straight rods measuring about 0.4 to 3 micrometer in tissues. On artificial culture media, when we inoculate and incubate on the artificial culture media, it appears as a coccoid and the filamentous in forms. Acid fastness, just we have discussed, acid fastness in nature due to the waxy envelope, that is the waxy cell wall. Zn technique is used for the staining of that particular cell wall. That is widely used for the identification of the Mycobacterium tuberculosis in the samples. Smears of the sputum or the sections of the tissue we can use or frequently used, that is the fluorochrome stains that is incorporated with the auramine or the rhodamine dye, and it is preferred in the clinical specimens, but the demerit it requires the fluorescence microscope, which is not available in every laboratory. So in the majority of the cases, the gene Lanson staining technique is used. So let us discover the pathogenesis. The pathogenesis is governed or directed by the different virulence factors of the bacterial components. Some bacteria produces the toxins like the endotoxin, exotoxins, and some bacteria, their cell wall, their proteins, their components acts as the different virulence factors. So likewise, in the case of the Mycobacterium tuberculosis, the cell wall that composed upon the lipids that include the mycolic acid, waxes, and the phosphatides, the cell wall of the Mycobacterium tuberculosis, and it also contained the muramyl dipeptide, which make the complex with the mycolic acid and cause the granuloma formation, which is the hallmark of the tuberculosis for the diagnosis. And the phospholipids, which is present in the cell wall, also induces the caseous necrosis that occurs in the lungs. Proteins, another component of the Mycobacterium tuberculosis is the proteins that elicit the tuberculin reaction. So in the primary infection, we can diagnose with the tuberculin test. Can also elicit the formation of the variety of the antibodies. So these proteins act as antigen, and this antigen will develop the corresponding antibodies by the plasma cells once the antigen has been entered in the host cell or the susceptible individuals. Another is the polysaccharide. It contains a variety of the polysaccharides and can also induce the immediate type of the hypersensitivity reactions. Can serve as an antigen in the reactions with the sera of the infected persons. So these are the acts as an antigen. Polysaccharide also acts as an antigen, and it is particularly used during the serological diagnosis. So these are the components of the virulence factors that we have discussed about the cell wall, proteins, and the polysaccharides. Let us join together that how it causes the disease, that is the pathogenesis. So we can observe here in the figure that here is the infected individuals. When the coughing or sneezing, he released a large number of the aerosol droplets that contains the Mycobacterium tuberculosis, and these aerosol droplets are inhaled by the susceptible individuals or the healthy individuals. And after the inhalation, the different innate mechanisms or the different primary defense mechanism, after the passing of the primary defense mechanism like the nasal mucus or the immunoglobulin A protease or the nasal hairs, it goes to the lungs. In the lungs, it will find the macrophages, alveolar macrophages, which is the army or the defense mechanism of our body, which will fight with that antigens or the bacteria. So these bacteria are engulfed by the macrophages, but macrophages cannot able to kill that Mycobacterium tuberculosis. So what happens? The Mycobacterium tuberculosis will multiply in the macrophages. And these macrophages, along with the macrophages, other cells like the fibroblast, like the dendritic cells, natural killer cells, these also accumulate, came to kill that bacteria, but they cannot able to do that. So they forms the granuloma. The macrophages, inside the macrophages, the Mycobacterium tuberculosis will multiply and they forms the early stage of the granuloma. This early stage of the granuloma is surrounded by the different immunological cells like the natural killer cells, dendritic cell, fibroblast, T cell, B cell, and form a foamy macrophages which trap the Mycobacterium tuberculosis inside it. And it is known as the caseous granuloma. So the caseous granuloma we can observe here. These are the Mycobacterium tuberculosis, the bacteria, and these are the cells. This is known as the caseum. And when the persons becomes the immunocompromised, then this will be the disintegrated. And after the disintegration, the bacteria will be released, and again the persons is able to transfer or make the another person to have the disease. Means the cycle is repeated. So here what happens after the formation of the granuloma, this granuloma, the caseous formation, cheese-like consistency. So this large number of this is the one, just this is the example of the one. Here, in the case of the lungs, many number of the granuloma will be formed in the alveoli of the lungs or in the lungs parenchyma or the epithelial cells. So here, just we have discussed that is the formation of the granuloma and the hallmark of the TB. Okay, the foamy macrophages that release their lipid content when they necrotize leading to the suggestion that the cheese-like structures. So that if it is a cheese-like structures, the bacteria will coalesce together or gather together or accretions to form the large number of the granuloma. So upon reactivation, we have discussed about the reactivations. The person become the immunocompromised. Reactivation, again the cycle will be the repeated, and the aerosol, contagious aerosol droplets after the coughing and the sneezing, the person is able to transfer to the another susceptible individuals. So what are the clinical features after the repetition of the continuous repetition of the cycles? What are the clinical features? So the primary infection is the reactivation. The primary infections is the lesions that is forms in the lymph nodes in the case of the lungs. And that lymph nodes, the formation of the lymph nodes of the massive suggestion that forms as a Ghon's lesion or the Ghon's complex. After the solidification of this massive suggestion, they calcify it and forms the Ghon's lesion that we can observe in the X-ray, chest X-ray of the infected individuals that is suffering from the tuberculosis. Another test we can go for the tuberculin test results becomes positive in the primary infection cases. And when we talk about the which part of the lung is infected, so in the case of the primary infections, the base of the lung is infected. But when it is reactivated, the apex of the lungs is being infected. And that is characterized by the tuberculosis, suggestions, and the fibrosis. So predisposing factor, what are the predisposing factors that trigger the development of the tuberculosis? So diabetes, high blood sugar. In the case of the diabetic patients, weakened immune system like the HIV or the AIDS, and other also malnourished individuals or the children's, tobacco use, cigarette smoking. So these are the predisposing factors that trigger the development of the tuberculosis. Common symptoms, what are the common symptoms that suffer or that shows by the infected individuals that is suffering from the tuberculosis? So in the case of the tuberculosis, person having the prolonged cough, sometimes with the blood that is known as the hemoptysis. So when the hemoptysis will occur, when the granuloma will disintegrate along with the different cells along the mycobacterium tuberculosis, there is the fibrosis. The blood tint, also a small amount of the blood will also appear in the sputum. Chest pain, weakness, fatigue, weight loss, fever, and the night sweats. So these are the most common symptoms that have been showed by the infected individuals. So TB usually affects the lungs, but it also affects the kidney, brain, spine, and the skin. So it is known as the extrapulmonary tuberculosis. So how is TB transmitted? The options are the inhalation of the infected droplets, inhalation of the fungal spores, inhalation of the dust and the vehicle smoke, inhalation of the bird guano. So many of the students are agree with the option A, inhalation of the infected droplets. Yes, right, great. So obviously it is transmitted by the infected droplets as we have read in the pathogenesis. Then we will summarize the topic. Microbial tuberculosis AFB causes TB, the most common infection of the lungs, transmitted by the infected respiratory droplets, forms the granuloma, which is a hallmark for the TB diagnosis. Primary infections that occurs at the base and the reactivation occurs at the apex of the lungs. Predisposing factors like the weakened immune system, malnourished, tobacco use, and the most common symptoms are the hemoptysis, prolonged cough, chest pain, weakness, weight loss, fever, and the night sweats. So we have completed the tuberculosis up to the morphology, pathogenesis, and the clinical features. So up to here, any questions? No any questions? Then thank you for the listening. Have a good day. Thank you.